Remember, proteins are made up of hundreds of amino acids. And so really what the code in the DNA is telling you to do is which amino acids to put together to build that protein. So let's go into this in a lot more detail now. The structure of DNA and how it works. Now, DNA is actually a double-stranded molecule. It's what we call a double helix. It's like a ladder that's been twisted up. And if you look at the ladder and the rungs of the ladder, those are bases in there. And there's 2 bases for each rung, and they're always in pairs, these chemical bases. The 4 bases involved are A, T, C, and G. A always goes with the T, and C always goes next to a G. They can go either way around, but it's always A and T and C and G, and we call this complementary base pairing. So the order of these bases in the gene determines which protein is made by the cell. But how exactly does that work? Well, let's look in detail at a process called protein synthesis. Let's take this strand of DNA. You only actually need 1/2 of the molecule. So this is, let's say we've got the double ladder, we've uncoiled it, to make it into a ladder, and we've ripped it in half. And this is just 1/2 of the DNA molecule here. And you can see the bases, T, A, C, C, A, G, C, T, A, T, C, C, etc. And the order of these bases is going to code for which amino acids we're going to select in order to build our protein. Now there's 20 different types of amino acids that occur in nature. Think about these like the building blocks of Lego. Think about your Lego models. Think about all the different types of Lego you've got. You've got little red squares, you've got longer thin ones, you've got flat ones. Let's say there's 20 different types of Lego block. The way you put them together can make thousands of different Lego models. This is like the instructions you get with your Lego model. You open it up, you turn the page, it tells you which block to get, turn the page, then get this block and put it on top of that block, turn the page, this block on top of that block, etc, etc, etc. And eventually, through all these different combinations of 20 different Lego bits, you build a very complex model. That's the same system here. Our DNA code are instructions. It tells you which amino acids to select, and then you can build a protein. We read the code in 3s, 3 bases at a time. So you'd initially start with, on this code, TAC. We call that a codon. Every 3 bases, these 3 bases is called a codon, and we read it in 3s, and that's why it's called the triplet code. So you take a codon, and that codon will determine which amino acid to pick. So let's say this gene is our example. Now in this gene there are 36 bases, but those are split up into 12 codons because we look at 3 at a time, remember. So this is going to code for 12 different amino acids. So there we go, we'll go TAC codes for this amino acid, CAG codes for this amino acid, CTA this one, etc, etc, etc. Each 3 bases codes for a particular amino acid. Sometimes it's the same amino acid because there's a lot more triplet codes or codons than there are amino acids. There's 64 different codons but only 20 amino acids. But eventually, as you can see, we have now assembled a chain of amino acids. These can then fold up and form a protein. However, actually, this process is a little bit more detailed than that, and you need to know about it in more detail. So it's actually split up into 2 stages, one called transcription and one called translation.